Hello everybody, this is the lecture number 10 part 2 about the simple stain. Simple stain. In the part 1 we explained the types of or kinds of bacterial stain: simple, compound, negative. Now we explain the procedure of simple stain to stain the bacteria. Simple stain because we use only one dye or only one reagent. We can use crystal violet or carbolfoxine, blue or red stain. Most the microbes have negative charge in on their surface. Why? Because the cell membrane and cell wall have lipid and lipopolysaccharide, so the charge of the bacteria will become negative, and therefore the bacteria will stained directly with the stain which have positive charge. Okay, microbes binds with the basic stains which have positive charge dye because the surfaces of the bacteria have lots of negative charge. بما أنه البكتيريا تحمل الشحنة السالبة بسبب وجود الليبيد، الدهون، ومجموعة الفوسفور أو الفوسفات الموجودة بالغشاء وجدارها الخلوي، لذلك فتحمل الشحنة السالبة. أما بعض الأنواع، يعني هي أكثر أنواع البكتيريا راح تكون تحمل الشحنة السالبة، لذلك فإنه سوف ترتبط مع الأصباغ اللي تحمل الشحنة الموجبة. Now, what are the equipment that must present to stain the bacteria with simple stain? What are the equipment or materials which must present in your lab to stain the bacteria by using simple stain? We need two staining rack, the materials in bacterial smear preparation, which are loop, slide, source of heat like Bunsen burner, bacterial growth either in liquid or on solid media, or any clinical sample. Okay, we need to either crystal violet or carbolfoxine, either or، أما أو صبغة زرقاء أو حمراء. filter paper, distilled water, 17% alcohol. The procedure: clean and dry the slide with 70% alcohol. Prepare the bacterial smear. Fix the bacterial smear. Firstly, we must dry the bacterial smear in the air and then fix the bacterial smear by passing the bacterial smear 3 times in Bunsen burner, in the flame of Bunsen burner. Put the slide on the staining rack. Stain the smear by using any simple stain, crystal or كاربولفوكسين. بعد تحضير المسحة البكتيرية نضع قطرات يا أما من الميثيلين بلو or كاربولفوكسين or كريستال فايوليت، أما صبغة زرقاء أو حمراء المتوفرة في مختبرات for 1 to 2 minutes. Wash the slide with tap water or distilled water and then let the bacterial smear after staining to dry in the air with filter paper and then examine the stained smear under low power and high power and then by using oil immersion to study the shape and aggregation of bacteria. Now we can watch this video about how can you stain the bacteria with simple stain. Firstly, you must draw circle in the bag of slide and then put it on the staining rack. Take 1 drop of distilled water by sterile loop or by Pasteur pipette, then sterilize the loop by red heat in the flame of Bunsen burner, then cool the loop and take a piece of isolated colony or only one isolated colony by the loop and mix the bacteria with the distilled water gently without touching the edge of a slide. Then let the slide to dry without any source of heating. After that, you must pass the bacteria 3 times on the flame of the Bunsen burner to fix and kill the bacteria. Then take either stain or then take only or add only stain, take only stain either red, for example carbolfoxin or neutral red or safranin. Let the stain about from 30 seconds to 1 minute. Wash the slide with the tap water or distilled water and then dry by using filter paper. Look the difference between stained and unstained smear. Examine the bacterial smear under the oil immersion.